Well, it looks like we're recording now. So hello, everybody. My name is Griffin Housen. I'm an undergraduate student here at the University of Windsor. Welcome to the third talk now in the virtual seminar series for undergraduate physics students. This week, our speaker is Dr. Stephen J. Rehse. His focus spans multiple subfields of physics, including experimental biomedical physics and AMO, atomic molecular optical physics. Specifically, he focuses on identifying pathogenic bacteria with laser-induced breakdown spectroscopy. So this talk and any of our previous talks are available on the UWindsor Physics YouTube channel. There will be time for questions at the end where you can unmute yourself and ask Dr. Rehse a question, or feel free to put your questions in the chat and either Dr. Rehse or I will read those aloud, and then he'll be able to offer an answer. So for now, I'll just have everybody mute themselves and put any questions in the chat, and I'll hand over the floor to Dr. Rehse to begin his presentation.
Okay. Thank you, Griffin. Okay, I have been made the host, so I will share my screen here. I thank everybody very much for attending. Sharing computer sound on a Friday. Okay, that should be up there. Griffin, does that look okay to you? I'll just confirm with our moderator that everything looks okay on his end.
Yep, yeah, I'm able to see that.
Okay, thank you. Then I will assume everybody else can. So thank you everybody for joining us on Friday. And for those of you who are watching this on delay on YouTube, thank you very much for tuning in to catch it after the fact. We greatly appreciate your involvement with us. So yeah, I'm going to talk today about the work that I've been doing for the last, over 10 years, I guess I would say, trying to use my background in atomic and optical spectroscopy and applying it into a very kind of clinical biomedical field, which is the diagnosis of pathogenic bacteria. So it's kind of an interesting story with a lot of interesting side roads on it that I hope you enjoy listening to. So let's go ahead and get started. We have to start by just recognizing that the University of Windsor sits on the traditional territory of the Three Fires Confederacy of the First Nations. And these are the Ojibwa, the Ottawa, and the Potawatomi. And the university respects the longstanding relationships we have with these First Nations peoples in this place, which is the 100-mile Windsor-Essex Peninsula and the Straits, or Les Détroits, of Detroit. We can start by actually looking at Les Détroits of Detroit here. So of course, here we are on Earth. We are all kind of huddled down in the southern part of Ontario. We can zoom in on that. And all of the work that I'm going to be describing today, and I show this, is kind of occurring within that flashing red circle that you have on the screen. And I'll explain that in a bit. But here you can kind of start to see Les Détroits of Detroit, the Straits kind of come through here and separate Canada from the United States. So if we zoom in a little bit more, so this is the reason that I show this, because this research story that I'm talking about today actually spans two countries. And from 2005 to 2011, I was a faculty member at Wayne State University, which was over here across the river. And in 2011, I moved to the University of Windsor. I guess I should say 2020 now, because I'm still here. So I moved across the river here. And in both places, I was working in different aspects of the same project. So the first question was just this: can this all optical technique of laser-induced breakdown spectroscopy, LIBS, and I'll definitely define all of that as I get going this talk, but can this all optical technique really identify bacteria in under a second? That was the research question, the hypothesis, if you will, that I was working on when I was at Wayne State. So my answer to that is yes. I do believe that this technique can really identify bacteria in under a second. And this has basically been, I would say, proven, or at least shown by us, in 8 publications spanning my time in these 2 places, in places like Applied Physics Letter and Journal of Applied Physics, Applied Optics, Applied Spectroscopy is a big one we publish in, Spectrochemica Acta, and others. And this has been confirmed by multiple other groups all over the world. This is certainly not just me saying this. I didn't come up with this idea. I was inspired by others, and I'm not the only person doing it, but it's pretty well consensus now that this technique actually does work for identifying bacteria. So let's see what happened then. I'm sorry. So if you want to know anything about it, if you have any interest in getting into the physics of it, I would suggest just starting with this review article that I wrote in 2019. It's kind of your one-stop shopping for all the information related to it. And then if you wanted to dive any deeper into it, which you probably wouldn't, you could go and find the references in there. But certainly you could start with this article here. So then I moved in 2011 to the University of Windsor, and the question changed. So having proven to my satisfaction that this technique really can identify bacteria, the question when I moved here kind of became, can we actually translate this technique into a convenient and easy to use test? And easy to use meaning for clinicians, doctors, nurses, health practitioners, something like that. So it's this idea that can we actually take something that works in the lab, so in theory, in the lab, I would say, can we move that into the clinical world? And to do that, we have to make a lot of advances in making it simpler, making it faster, and making it more automated. And that's been the focus of my research since I've been here at the University of Windsor. And we'll see some of the things that we've done in there. So here's going to be the outline of the talk. I'm just going to give an introduction to our method. I'm going to talk in general about this thing that we call LIBS, which is laser-induced breakdown spectroscopy. And then I'll talk about some of its advantages that I think it has over competing methods. And then I'll get into the nitty-gritty of what I do for my work, the biomedical applications, and finish up with some concluding thoughts and hopefully take some questions from you all. That would be great. So we start off with this idea of a laser-induced plasma. So what is a laser-induced plasma? Well, a laser-induced plasma is a plasma that's induced by lasers, I guess. The idea is this is a plasma. So it's a hot collection of free atoms, ions, and free electrons. And it's created by an incident laser pulse. So what I'm showing here is a cartoon of an incident laser pulse coming from the left to the right here. And it's incident on this target. And basically the energy of that laser is converted into the thermal energy of this plasma. All right, so this is actually a picture taken in our vacuum apparatus in our lab. That bright glow, this was taken with a cell phone camera, so you definitely don't need special equipment to see this. It's very, very bright. You can see it with your eye. And this actually is a rare earth metal target for different reasons. We wanted to look at the rare earth metals. This type of analysis can be done with a whole host of different lasers. So they have to be pulsed. So lasers can be nanosecond, picosecond, or femtosecond pulse length and duration. It's been shown to work in all of those. All of the work I'm going to talk about today, we do with the nanosecond lasers in our lab. The threshold irradiance does have to exceed a certain value. That's what the idea of the threshold is. So a very low power laser would not do it. You need a certain amount of pulse energy. You need to focus the laser down into a spot to overcome this threshold irradiance, which would overcome the breakdown threshold. And then this breakdown event will happen and then a plasma will form. I'm going to get into the physics of all this in a very kind of cursory way in just a second. So let's get into that kind of just cursory discussion, the LIBS primer, I like to call it. So 4 slides usually that describe how exactly LIBS is done. It's really kind of a 4-step process. And the first step is going to be this laser has to interact with the target. Now the target in this case is going to be this green box. It's just a cartoon, of course, you understand that. And it's going to be a solid, but this technique works equally well on liquids and even gases. So it doesn't have to be a solid target like a piece of steel. It can just be a cuvette of some gas. And we've done experiments like that in my lab as well. But I think it's easiest for you to understand if you think about it as a solid target. That's the easiest kind of experimental and theoretical model. So we bring in this pulse laser. It's going to be coming from the top of the screen to the bottom. And the whole process is initiated as the energy of that laser pulse starts to become absorbed into the target. And so that's going to look something like this. So the laser pulse, again, so pretend this is a 10 nanosecond pulse duration laser pulse. The leading edge of the laser pulse starts to impact onto the target, and the energy of that pulse cannot be transmitted through it, so it starts to be absorbed into the target material. These are all thermal processes, so they happen on the picosecond timescale, so much, much shorter than the duration of the pulse. And we're indicating that absorption of the pulse energy by this kind of dark green oval here. This absorbed energy, what's it going to do, right? You have an electromagnetic energy. Energy is always conserved in physics. The first rule of physics is conservation of energy. So that absorbed electromagnetic energy has to be converted into heat energy, thermal energy. And that thermal energy is going to result in vaporization of the sample. And when you do that, that's called ablation or laser ablation. It's the direct boiling off of atoms and materials from a solid surface. So it's going to start ablating, boiling off atoms when the temperature reaches the boiling point of the material. And all materials have a boiling point, even pieces of metal. And so we're essentially boiling our targets, and those atoms start popping off. So as those atoms and nuggets and other clusters and things start popping off the surface, that's what we call the removal of particulate matter. So that's going to lead to the formation of a vapor cloud above the surface. And this laser pulse is still illuminating the sample because this is all happening in picosecond timescales, and the laser pulse is like 10 nanoseconds long. So this is the ablation step. So it kind of looks like this. So you see what's happened. The material has been removed. You're starting to form a crater, and all the mass from that crater is now contained within a cloud of vapor above the surface. And there's a bunch of physical processes that are going on to make that vapor cloud: atomization, sublimation, fragmentation, melting. So these are all thermodynamic processes that are occurring right at the surface of this material. But what you wind up with is this light green vapor cloud that I'm showing here. It might have some clusters in it that's being illuminated continually by this laser pulse coming from the top of the screen. When you do this ablation event on different targets, this is kind of what it looks like. So these are all, I believe, except for the lower right-hand corner, these are all scanning electron micrographs of shots taken in our lab, of laser shots on the nitrocellulose filter that we use to sample bacteria. I had a student who was working on human fingernail studies, which is shown here. And here's the laser shots on a piece of steel that we use for calibration. And you can see that sometimes it behaves beautifully and forms nice little kind of almost hemispherical craters. And then sometimes it works really, really badly, like on the human fingernail. So this is a matrix effect, which tells you that it does have a lot to do with what does that energy do once it goes into the material as it tries to boil that material off. But in general, it can behave very, very well. And you see these nice kind of 60 to anywhere from 50 to 100 micron diameter circular areas where we've removed little bits of the material. That's called ablation. So the third step is the formation of that plasma. It's a laser-induced plasma, so you have to initiate the plasma, and we call that event breakdown. So in the breakdown event, remember this laser pulse is continually illuminating the vapor plume from above. The vapor is expanding and cooling, so it condenses into kind of sub-micrometer droplet size. And these droplets and other particulates in that vapor lead to absorption and very strong scattering of the laser beam, which very, very quickly leads to extremely strong heating of the vapor cloud, eventually ionization, and then plasma formation. So it kind of looks like this. So you can see that that laser pulse, which is still illuminating from the top, it's not reaching the surface anymore because now it's actually being absorbed by the vapor. This is a thing that we call plasma shielding in my field. It means that all on its own, the plasma reaches a critical density where it's now absorbing all of the radiation and you are no longer illuminating the surface in any way. When that happens, this is a very, very kind of almost exponential increase in the thermodynamic properties of that plasma. And it kind of leads to a very violent event that we call the electrical breakdown and plasma formation. So electrical breakdown is exactly what happens when a lightning bolt flashes, and you've all seen lightning in the sky. That's a breakdown event, right? There's a high potential difference between the ground and a cloud. It just goes, and it goes almost instantaneously, opening up an ion channel where electrical current can flow. And that lightning channel is a plasma. So it's very, very similar to our laser-induced plasmas, what a lightning bolt looks like, or also what the spark in a spark plug looks like as well, same thing. In fact, Physics Today in May 2007 had this wonderful cover story that I always like to talk about in my talks, and only a physicist would understand this pun. Looking at who's on the line, it's probably only Professor Rangan, but I'm sure she is chuckling to herself right there because their pun is, this is a stark look at plasma breakdown. And for anyone who's taken or taught quantum mechanics, they know what the stark effect is in a high electron density plasma region. So that's a bad physicist pun. But nonetheless, they were trying to study this breakdown event. And in this picture, you can see between two highly charged surfaces, you can see the breakdown occurring in between. And it's intrinsically a stochastic process, which means you don't really know when it's going to go, and you can't control it exactly. And that's one of the problems with the advantage of our technique is speed, but one of the problems is there's intrinsic shot noise due to the stochastic nature of the plasma formation, which you just can't control, and no one can control. So when that breakdown event occurs, you see that bright flash. I've showed you the pictures of it. It's accompanied by a supersonic shock wave, which is a quite loud event in our lab. And I'm showing that shock wave expanding away from the cloud, and everyone in our lab can hear it very clearly. In fact, Jeremy will be, my graduate student Jeremy Marvin is on the line, and in his master's defense, which is coming up in a couple weeks, he's talked about a potential future work is to actually analyze the sound that these plasmas make as a diagnostic for how energetic that breakdown process is. That's just a super, super cool idea that we're hoping to get into in our lab. We haven't started doing that yet. We're still in step 3. This is very, very early on. So the laser pulse is done illuminating it, but I would say we're in the first 1 to 25 nanoseconds of the life of this plasma. It's very, very hot. It's roughly anywhere 50,000 Kelvin to 100,000 Kelvin with high electron densities. It's really hot, hotter than the surface of the sun. And so all the light that you saw in that picture, the light coming out of that plasma is not specific to the atoms in that plasma. It's dominated by two non-specific sources of illumination, which would be continuum emission, which is as there's a ton of ions and free electrons in this plasma. And you all know about bound-bound transitions within an atom. When an atom makes a jump from one energy level to another, it gives off a photon specific to the gap between those energy levels. But when an ion grabs a free atom, a free electron out of the continuum, it can come in and give off a wavelength of any, radiation of any wavelength. So that's continuum emission. And then there's also Bremsstrahlung, which is the breaking radiation due to the presence of all these charged particles and ions and electrons, and that also is non-specific. So it's just a bunch of white light, I would say, coming out of the plasma, which doesn't tell you anything about anything. Looks cool, but there's no information in it. So let's take a look at what those plasmas actually look like. So I'm going to show you just a very short video that was taken in our lab where you're going to actually see in real time the analysis of a piece of steel. So you'll be able to see the laser-induced plasma and the analysis. So let's go.
All right, are you ready?
I'm ready. Oh, wow.
And there you can see it lighting up. And then here's the plasma. And you'll hear this spark. Alright, now let's have a look what we got here.
Wow, that's really cool.
Alright, looks like we found gold.
Oh, really?
No, it's just steel.
Oh, okay.
So that's essentially what we do in our lab. We shoot things with the laser, it gives off that flash of light, and then we look at it on the computer, and Jeremy was thinking he found gold in the piece of steel, but there was no gold, it was just steel. But you can kind of see a spectrum on the screen. I'll talk about this, but that light that comes out of it tells us everything that is in the material we just shot, and then we're using it for different types of applications. If you want to see any of the rest of this video, looks like everybody on this call right now has seen it, but I hope you YouTube watchers at home, after you get done watching this talk, you can go and watch it's a 4-minute video shot and completely done, narrated and shot by the students in our lab. Just go to YouTube and search for UWindsor Physics Rehse, and you'll find this cool 4-minute video that tells everything that we're doing in our lab, and that was a student-produced production, and they did a very, very good job. So thank you to Jeremy and Sydney who appeared in that previous video, and these are my current students, Emma and Sydney. This is just a screen grab. You see Emma sitting in front of our biohood making some bacteria samples. So they did a very good job on that. It's very funny. So the fourth step then is, because all of that continuum emission doesn't tell us what's in the target, we have to wait. So the step 4 is expansion and element specific emission that comes at some delay time after the plasma is formed. So the dynamic evolution of the plasma plume is characterized by a very fast expansion. So you can see this plasma growing, and it cools. And once it cools to about 10,000 Kelvin in about a microsecond, if we wait that long, it's cool enough where now we're not seeing those other broadband sources of emission. We're actually seeing spontaneous emission as the atoms and ions in that plasma decay to the ground state. And that spontaneous emission is going to, of course, be unique to transitions within the atoms. So this is going to be emission only from atoms or ions that were contained in the material that was just ablated, or perhaps in the gas cloud above it. And that's really what we want to do. So you see these different very specific colors being given off here. What we want to do is collect those colors and analyze them. So this is kind of what it looks like. The point is, if you take this light, which is very bright, and you put it into the proper instrument, you can do something with it. So here's the laser-induced plasma. So if any of you guys know anything about plasmas, there's lots of different kinds of plasmas. From the center of the sun is a plasma, a laser plasma. These are our laser-induced plasmas. Here's the photosphere of the sun. So we're in this range down here where, it's not the hottest of plasmas. It kind of spans, 0.1 to 10 eV, somewhere around 1 eV might be right, but it's quite high in electron density compared to a lot of the other plasmas. So we have about a 5 to 50,000 Kelvin with 10 to the 17 to 10 to the 20 free electrons per cubic centimeter. So this is just for people who know a little bit about plasmas. With that plasma, because it's hot, it's glowing, it's giving off light. If it's glowing and giving off light, you can do spectroscopy on that. And I am primarily a spectroscopist now, so that's what I would do. Take that light, put it into an instrument, break it into its constituent colors, and then see what that light tells me about the target or about the laser target interaction. So it kind of looks like this. You can do a little experiment at home. Here's LIBS on water, here's LIBS on copper, here's LIBS on steel. Steel, copper, water. Water, copper, steel. I'm going to go through them quick. If you guys look at it quickly, can you tell that those are different colors, particularly between the steel and the copper? Purple, blue, purple, blue. Your eye can tell the difference between them. Congratulations, you've just been doing spectroscopy. Spectroscopy is the identification of that material based on the color. Now, you don't know exactly what colors this is, but even your eye is a good enough instrument to analytically know that this material is different than this material just due to the color of that. Of course, we don't use our eyes. We use really expensive equipment to analyze the color of these plasmas, but that's essentially what it comes down to. Can we really analyze that color, quantify it, measure it, and get some usable information out of it? But that's the idea of the spectroscopy. So our goal when we shoot that laser is actually to create an optically thin plasma. And that's just a phrase that means that all the light that gets released by the atoms actually makes it out of the plasma. So it can't be so hot and so dense like a star, because none of the light makes it out of there. So it needs to be thin. It needs to be in thermodynamic equilibrium, or local thermodynamic equilibrium. And we hope that the elemental composition of the plasma is the same as the target, because we're analyzing the plasma, not the target, but we assume that those are similar. If that's achieved, then if we can measure the intensities of the observed spectral lines, so here's like a spectrum of steel, what Jeremy was looking at, so that see the height of all these lines, that's the intensity of those lines. We know where they are. We don't measure that. We don't care where they are. We know exactly where they are. That tells us what element it is. But if we can measure those intensities, that intensity can be related to the relative concentrations of those elements in the material, sometimes even the absolute concentrations. The problem is typically these conditions are only met approximately, meaning we're not really, that plasma is not all completely in LTE. It's not always completely optically thin. So sometimes we have to make some compromises, but we do what we can with the plasmas we get. So here's a time graph of what my experiment looks like. So in the first, start a stopwatch, in the first 0 to 10 nanoseconds, the LIBS laser pulse comes in. During that time, the emission from the plasma is going up, 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 up. After the pulse is done, it's not absorbing any more energy, and it's probably just getting dimmer and dimmer and dimmer and dimmer and dimmer all the way out. We wait till about 1 microsecond, and then we look at the plasma for about 10 microseconds. So we have a spectrometer camera that allows us to do gating of our observation of the plasma. So we just call that the gate delay. But when you do a time resolved spectroscopy, that is, we look at the elemental emission lines in the plasma out here, not time averaged over the entire process, which would be a mess. We wait a little bit to allow them to get spectrally narrow, to let the plasma cool, to let the electron density come down. When you do that, that's called laser-induced breakdown spectroscopy. So it's a bit more than just firing a pulse of laser into a target and looking at the light, because it does denote that we have that time gating possibility. And that's what we call LIBS. So I'll be using this acronym LIBS for the rest of the talk, laser-induced breakdown spectroscopy. But after that little primer, I think you all know what the phrase laser-induced means, and now you know what breakdown means, and you kind of know what spectroscopy means. So this is not a new technique. In 1960, Myman came up with the first ruby laser. So there's a picture of like what a ruby laser would kind of look like. Almost immediately in 1962, LIBS was born when a paper was published detecting a spectrum from ruby laser-induced plasma. So right away after they had a pulse laser, the first thing they did was shoot it into things, focus it down, and then look at the light coming out of it. So it took all of 2 years for that to be born. In 64, a paper appeared that was the first direct spectrochemical analysis by LIBS. So spectrochemical means I'm able to determine the chemical composition based on the spectrum of material. And in 65 was the first theoretical model for what's actually going on in that laser breakdown process. So it's kind of an old technique. I mean, it's been known for many, many decades, but interestingly enough, nobody was using it until about the 90s, right? So it was invented in 62, 64. So what I'm showing here is this is a common trick that physicists like to do, which is you just take the field that you're publishing in, like Dr. Rao could have done that on his spin ices, which is a new thing, and you just search one of our online, through the library, one of the online databases. In this case, it was the Web of Science. And you just say, how many publications appeared with that phrase or this phrase in the title? And you can see that since about 1990, it's been really having this nice increase. And this was led to all of a sudden in the 90s, it was the improvement of detectors, really fast cameras that allow good gating, and spectrometers that are kind of smaller and portable and cheaper, where a lot of people could do things with them. So it was known for 30 years, but it took 30 years for the technology of spectrometers to catch up to it to make it actually useful. And now, this is a very steep rise in the number of papers. Why such a rise? In the number of papers? Because it has a lot of applications, lots of applications, and lots of advantages. I could just spend a whole talk talking about the different applications, which are really, really fascinating. The interesting thing about all the applications, so I'm co-supervising a student over in the School of the Environment, and he's using it to look for gold in rocks. And one of my students was looking for zinc in human fingernails. And our current project is looking at bacteria in clinical specimens. And people are using it to look for explosives and looking at to quantify archaeological artifacts and conflict gemstones. So many applications, but in all the applications, as I thought about all of them, all the papers that have appeared on LIBS, if they're not a theoretical study, I've determined that all the papers are attempting to do one of two things. You are either attempting to quantify the amount or concentration of some element by analyzing a peak intensity. So that is, you just look at the size of that peak, and if you are careful, and it's almost doing it's called an analytic chemistry, you relate the intensity of that peak, the size of that peak, to how much stuff was in the target. And I've done studies like this. We were trying to determine how much zinc is in a human fingernail. And is that related to how much zinc you have in your diet? This is a study done when I was at Wayne State, where we were looking at lead collected from the dirt on the side of the road. The United States used leaded gasoline for decades and decades. So it turns out that all the dirt, this is just a dirt sample taken right off of I-75. All that dirt is still contaminated with lead, because this literally came off the side of the road, and our technique can determine the lead presence in it. So there you go. So you're either doing that, Or I don't care about the intensity of that peak, but can I look at where all these peaks occur? And can I then use that as a unique pattern to identify a target based on that unique elemental composition, where I don't care what's actually in it, but is it unique enough that when I shoot it, I get this pattern every time and it becomes a fingerprint. Sometimes we call it a spectral fingerprint, or you can think of it as like a spectral barcode, like the UPC code on any product you buy, is the intensity and where those lines occur. If it's unique for a given thing, then all I have to do is shoot it, and then I can always tell that that thing is the thing that I'm looking for. And that's what we're really doing in all of our bacteria studies that I'll talk about today. I don't really care what elements are present in the bacteria. I don't really care how big those lines are. I just need them to be the same every time so I can uniquely identify a bacterial cell. But everything in LIBS is going to be one of these two applications. People like to use the LIBS because it does have advantages over other methods. One of its advantages is it's very sensitive to almost all the elements. We call it multi-element sensitive. There's really no other spectral technique that can detect all of these elements without bias. Particularly the light elements are not detected by X-ray fluorescence, which is the main competing technology. It doesn't detect the light elements very well. All of the elements can be detected via LIBS without bias, and that's a big, big advantage. Because we focus that laser down to a small spot, in our case, it's about 50 microns. In the case of some labs, they can make it 1 to 2 to 3 microns. You get exquisite point sampling. We are not the most sensitive techniques. There are certainly elemental techniques that can measure part per billion and part per trillion concentrations if you have like poisons in you. We can't do that at all. But to do that, they need a big amount of sample, and they dissolve the whole thing. They digest it in acid, and then they put it into their apparatus. We can fire our laser and get these point measurements. So what you're seeing here is a LIBS surface map of a kidney slice. So these are my colleagues, Vincent Motoros in Paris, and they took a mouse and they fed it gadolinium intravenously, and then the mouse was sacrificed, and they did slices through the kidney. And these are all, I don't know how many million laser shots, but then they took that shot and they plot it with a color depending on the intensity of the gadolinium and sodium lines that they saw. So this is not a photograph. This is a LIBS image where every pixel is just color coded to the intensity of an elemental emission line they saw. But you can see this exquisite kind of resolution on that kidney slice, which is very cool. And because we only remove micrograms to nanograms of material, when they do that, it makes a very, very thin crater. But that means you can just sit in one location and start to dig deep. So you can get that same spectral or spatial resolution in depths through the material. And that's what you're showing here. The same type of slice of a kidney, but then in progressively deeper layers. So it's truly three-dimensional elemental analysis. And none of the other techniques have anything like that because they tend to digest the entire sample. So that's a really big advantage of it. My biggest advantage is that we can measure concentrations of 1 to 100 ppm, which I said is not the best in the world, but we can get it in about a second using standard LIBS apparatus. So it's fast. It's really fast. And that speed of determination is really the thing that I'm chasing to try to make it clinically relevant. Because it can be done fast and you don't have to take it in the lab, it can be portable. And so first responder CBRNE prototypes have been built. So CBRNE is what this guy is wearing. That's a CBRNE suit. So it's chemical, biological, radiological, nuclear, and explosive. So these are the hazmat guys who come out, and he's testing an unknown white powder. So you can see that he's carrying everything in a backpack with a handheld delivery device. Laser Spark would be there. And we are trying to come up with computerized algorithms which will autonomously characterize that plasma emission. And then it makes decisions on the threat level of that target and conveys it to the operator via a head-up display. And that all can be done in about a second. And there's nothing else like that on the market. Here's another example of a different company's idea for a man-portable LIBS system. You can go man-portable, you can go high energy. So a lot of this initial work was done by the military in the United States. So this is a high energy remote system. You can see they've just mounted the 2 lasers, same second generation apparatus here, Mark I apparatus here, 2 lasers and a spectrometer on a big Schmidt-Cassegrain telescope, which in this picture is mounted in the back of a truck, and they're shooting it out at this target right here. Off in the distance, you can see a little box into which they've rubbed some explosive residue. So they're developing it for the Army to mount on Humvees to do roadside bomb detection at what we call standoff distances. You can do that too. You can get commercial benchtop systems. So if we ever get our bacteria stuff to work, I envision a machine that looks like this, very at home in a clinical lab or a doctor's office or nurse's office. It would go right in there, little doors, all the samples go in. You can do that. You can even do handheld. So like I said, I was co-supervising a graduate student in the School of Environment for his application out in Western Canada with gold prospecting. He was using this LIBS from SciAps. So this is a handheld LIBS device. It kind of does everything I was talking about in this nice handheld device. And usually they have now kind of cell phone app driven analysis right on the back of it. So you can just upload that right to your phone and do everything on an app, which is super, super cool, but definitely man portable, which is great. And most importantly for my field, to prove that this stuff actually works, a system has gone to Mars. So this is the Mars Curiosity rover. It is shooting out this is an artist's illustration, but it is shooting out its laser. This is a LIBS laser onto a nearby rock. And it allows, on the Mars rover, it allows up to 7 meter standoff analysis of rocks, which is unlike any of the other instrument packages on Curiosity, which require rover to be in contact with the item. And oftentimes it brings up a sample, then brings it on board for direct analysis. LIBS is the only one that is able to make analysis at distance, which is good because the rover does not actually move that much. And for it to be able to move its masthead around and shoot at targets is a big, big advantage. This is actually what the Mars system looks like. This is called ChemCam. So ChemCam is a high resolution camera imager and LIBS system. So this is the masthead part. So ChemCam is this thing up here, looks like the head of the robot. And then this is what the, actual LIBS unit looks like inside ChemCam. This is developed by my colleagues at Los Alamos. This is the first selfie on Mars. So this actually is a picture. So this is the first selfie ever taken on Mars. This is a self photo of Curiosity. It's taken from a camera on the end of its reacher arm looking back at itself. This is the ChemCam where the LIBS instrument is. And somewhere back here, actually right back here, these are actually LIBS calibration standards. So I was pointing this out to my research group a while ago. You see a very small series of dots there. These are dots that the LIBS laser looks back, fires at to calibrate the spectrometer. It's a bunch of known rock samples, and then it can go off and start to shoot Mars rocks. Of course, my lab doesn't look anything like the Curiosity rover. My lab looks something like this. So it looks like a physics lab. It's got equipment scattered all over. I've got about 2 or 3 different LIBS setups on these different tables. This is Bo Greaves, my last undergraduate thesis student, who was working on one of these experiments. So you can see it's a bunch of lasers. They're very, very big. They're not handheld. They're not portable. They're bolted to a table. But this allows us to do anything we want with the experiment. So we're not engineers. We're not trying to build a small box yet. But you can see the size of it. My lasers are quite big. My spectrometers are quite big as well. But this is kind of what a LIBS experiment would look like in the lab. And this is what a student looks like shooting it. So this is my student Laura. She's wearing her regulation laser safety glasses. She's kind of steering a laser spot on this monitor to her fingernail so she shoots in the right place. And this is just our ablation chamber. Jeremy was showing this before. We shoot everything in argon. So it's got a little door. We put our samples in there. We close the door. We flush it with argon, and then we take our data. Here's just another close-up of that. This is my student Cyrus. That was a picture at Wayne State. Here's a picture from a couple years ago in my University of Windsor lab, just kind of showing it takes up an entire optical table, but there's a couple experiments going on here. Okay, so let's just get into the application I'm most interested in, biomedical applications. I really do consider it a new paradigm for rapid pathogen identification. Of course, with the COVID, when have we ever in our lifetime ever been more interested in rapid pathogen identification? I would argue never. But they all chase down the same old roads. There's not a whole lot new going on. So sometimes it's good to think outside the box and say, we have new ways, and using a laser is a very, very new way of doing it. It's not a wet chemical way. I got interested in this, and my whole field got jump-started by the September 11th bioterrorism events, right? So the first incident of really bioterrorism in the world, which was a real anthrax biological weapon, was mailed to a bunch of places in the United States, including a senator in the United States Capitol. And basically what they realized is that whether you have a powder residue that could be potentially fatal anthrax or a watch glass full of cornstarch, when this happened, the powers that be, and that meaning specifically the people who fund us and give us money to do things, they looked around and said, hey, do we have any technology that those first responder hazmat guys can carry that when they go into the United States Senate building, they can immediately tell that that's anthrax or it's cornstarch? And the answer was a big no. We have nothing like that. And the people who write the check said, really? You have nothing like that? And we said, no, really. We can take a sample and we can take it to the lab and we can let you know in 2 weeks what it was. And they said, well, that's not going to work. So who can figure out how to do this? And we said, we can do that. And so all the money started to flow. And so the first papers took off in 2003. It was a direct response to September 11th. Now, you may not be scared of bioterrorism events. To be honest, I'm not scared of bioterrorism events either. Here's my take-home message on that. You don't have to be scared about bioterrorism events, because what people have realized is they don't need anthrax to shut down government buildings. All you need is a mysterious white powder, and the hazmat people have to respond as if it is anthrax. So the same thing happens. They've accomplished their goal of disrupting and shutting things down without having the real event. So this is, I just take my lead-ins to my colloquia come from the Windsor Star front page. November 10th, November 2010, mysterious powder investigated. I love this. So here's the guys in their famous white hazmat suits, no LIBS instruments. Hazardous materials team with Windsor Essex Fires takes readings and samples from the contents of a recycling box, blah, blah, blah. It was a pencil case containing a white powder. What is it? We don't know. Look at the last line. It's been taken to a laboratory in Etobicoke for testing. And guess what? You never see a follow-up story telling you what it was. It goes off to the lab for testing. Yeah, we'll let you know in a week or two. Well, great. I guess as a physicist, I say, surely we can do better than that. Surely we can do better than that. Remains to be seen, I guess. Here's another event. Suspicious powder scare empties building. Again, here's your firefighters. Here's 2012. Suspicious white powder, Canada Post. Everybody evacuate. Everybody shut down. How much does this cost? $100,000, maybe $50,000. Here's another event. Suspicious powder at National Bank. Not dangerous, but here's these guys in their ubiquitous white coats. Again, suspicious white powder. So why do we not have a handheld device that can right on the scene tell them what this stuff is? We don't have it yet. That's kind of what I'm trying to work on. So why is it? Why do we not have a technology? Although we can certainly identify bacteria in a lab, it looks like this when they do it. Why can't we do it right on the scene and say, oh yeah, that's a bacterium? Well, when you talk about foodborne contamination, why do people still get sick and die of food poisoning? The answer is, from this reference, it's pretty well accepted that the microbiological expertise, meaning you need like PhDs in microbiology, and cost required to perform these identifications preclude their common use as a screening mechanism. So they're just too hard and too expensive to do on every piece of sample that could be contaminated. So we don't use it, which is why people still get sick. If we had something that was cheaper and required no expertise, now we're solving the problem. But this was the state of the art. So here was our thought. Here's how we do it. So we're simulating, say a urinary tract infection. So you could get a urine sample, or sepsis, which is bacteria in a blood sample, or we're simulating a contaminated bedside, contaminated telephone pole. This could, telephone on the side of your bed, or this could be a swab that comes out of your nose from the hospital. So we can simulate those things that come off the surface. We swab it up, and we collect the bacteria off the swab. That's what doctors use when you have an infection. And we're trying to get the bacteria shaken off of that in a vortex mixer and deposited on a filter via a centrifuge. If you give me a fluid sample, I can just centrifuge that directly through this device that we built, and then the bacteria deposited on this filter, and we can sample on that. So that centrifuge insert looks like this. So this was a master's project of one of my students. So he designed and had this 3D printed. There's nothing like this in the world. I can tell you, I have people send me emails saying, hey, can we get one of those? I say, no. I only have them, but I'm happy to share my plans with them. And so people are trying to start to make those. They don't make them anywhere. We invented this. You would think it would exist, but it didn't. It's really strange. It goes in a standard centrifuge tube, which goes inside a standard centrifuge. We put all of our bacteria containing stuff up here. There's a paper filter. It's nitrocellulose, but it feels like paper. It screws into the bottom. You run it through the centrifuge here. Centrifuge has like 2,500 Gs. All the water gets pulled through. The bacteria cannot make it through the pores of this filter medium. So the bacteria is sitting on the top. We unscrew it, open it back up, take this filter out, shoot it with our laser. That's what we do. Very clever idea. So when we do that, we take that filter paper, we mount it on a piece of steel just because it keeps it flat, makes it rigid. We have a magnetic base that holds it in place and everything. It's really nice. And then here's our laser spark on the surface of that filter. It looks like that. When we do that, here is a LIBS spectrum. This is what we see. So when we shoot bacteria, we see a strong carbon signal. Bacteria are mostly carbon, oxygen, hydrogen, and nitrogen. We don't look at any of those elements except for carbon. We don't look at oxygen, we don't look at hydrogen, because that's all in the water, and the water's not specific. We look at inorganic composition. We look at calcium and magnesium, sodium, sometimes potassium. Phosphorus is a big one because bacteria have a lot of phospholipids. These are the elements that are unique to the bacterium, not oxygen, not hydrogen, not nitrogen. People say, oh, Steve, aren't bacteria mostly the same? I go, yeah, they are. Aren't they mostly water? I say, yeah, they are. They're 99% the same. And guess what? That 99% we don't look at, because we don't look at lines from oxygen. We don't look at lines from hydrogen. We don't look at lines from nitrogen. That's the stuff that's the same. What's not the same is how many calcium divalent calcium ions does it have in its outer membrane, which holds the bacterial cell together. That's very unique and very specific. That's what we base our analysis on. So the ratios of these peaks tells us that this spectrum comes from a particular type of bacteria, even though they all look the same. So my student did a scan of the LIBS laser across that filter. Yeah, they deposit pretty uniformly. It's pretty nice. When we did that, we could see about 90,000 cells. We call those colony forming units. So every laser shot was shooting 90,000 cells, which is way too many, but at least it works in principle. My next student had an even better idea. This is Allie's idea. Can we just take a small device with a little cone, plunk this guy in the middle of it, and then when the water is pulled through the centrifuge, it's not spread all over the filter. It's only spread right underneath this 1 millimeter hole. Well, that's a pretty good idea. When she did it, that worked really well. So now instead of being spread all over this filter, all the same bacteria are concentrated in the center. And here's a image you can see of that filter, and you can see each of these dots is a laser shot as we move our laser across the sample, and the bacteria are only right in the middle. So it acts to concentrate a very low number of bacteria into a small spot. Even this is way bigger than we need because, like I said, our laser spot is only about 100 microns in diameter. If we could concentrate all the bacteria into a 100 micron spot, that might be ultimately the way we want to do it, but that's harder than it sounds, but that would be the idea. So here's some scanning electron microscope images of what we see when we do that. So when we've deposited Staphylococcus on the filter, here's the layer of the Staphylococcus. You can see all these cracks in it. It forms this like dry desert lake bed view. It dries up and then it cracks, but that's all bacteria sitting around in there. And then sometimes we treat it with a chemical to make it lay better, and then you see the cracks didn't form. But you can look right at the edge of where that cone tip was, and here you can see no bacteria, and if you can see a little transition here, this is all bacteria here. This is maybe a better image. Here's the filter itself, and then it kind of transitions. It's a sharp edge. This is all bacteria, no bacteria here. So this tells us this is working really well. Here's an image where you can actually start to see the clumps of these Staphylococcus bacteria in this kind of mucoid film here on the filter. When we did this, the limits of detection are pretty good. For all elements, it's about 10,000 cells, but if you just look like at the calcium and magnesium, it only takes about 1,000 or 1,700 cells to actually see that. So this actually is working much, much better. So the limits of detection are better, but the number of cells required to get this is way, way lower because we're concentrating them all in one middle spot. Beautiful, works beautiful. we came up with another idea that we're trying to go to start doing blood studies, which is if we built this thing. So again, this is the first graduate student's idea. This is brilliant. If one piece can screw into it, why not have a second piece which screws into that? And then we put 2 filters of different sizes in there. This is a 5 micron pore size filter. This is a 0.45 micron pore size filter. So we put a bunch of stuff up top. The big stuff will get caught on that first filter. The small stuff will go through, get caught on the lower filter, and then just the water or liquid goes through to the bottom. And this was tested with tungsten powder because we just wanted to test it with something easy. And we put tungsten in there, and sure enough, the tungsten, which is about 12 micron, was all caught on the 5 micron filter. None of it passed through to the 0.45 micron filter. And there was bacteria mixed with the tungsten, and about 80% of the bacteria did make it through that first filter to get collected on here, and we were able to shoot it. Worked great. The reason we're interested in that is this is what happens when you get sepsis. So that's bacteria in your blood, which you should never have. If you have bacteria in your blood, this is a very fatal, very dangerous, almost fatal disorder. So this is a red blood cell, which is huge. And this is bacteria, which are small. So red blood cells are about 10 microns. So if we could demonstrate this in blood, we could run this blood sample through that thing that we just demonstrated. The red blood cells would be caught on the top filter. Only the bacteria pass through, and then we shoot and we see the bacteria. So that works pretty good. So does it actually work? Yeah, it works. So how unique is unique? Well, using the LIBS spectrum, we've done a lot of analysis of, this is a test of 4 different genera, which is the genus of a bacterium. So E. coli and staph and strep and mycobacterium. And using 2 different computerized analysis ways. So if you looked at those spectra by eye, you would never tell the difference. I will freely admit that. But a computer can. So we use things called chemometric algorithms, machine learning algorithms. We can teach the computer to recognize these patterns, to train them to recognize the patterns. And then when we encounter an unknown thing, we ask that trained algorithm, what do you think this is? And then it will, in a statistical way, match it up with the pattern it thinks it is. This is a really, really important new thing going on, this machine learning. I know that Dr. Maev in our department is doing the exact same types of things in his ultrasound analysis out at the IDIR. I know Dr. Zhao is doing the exact same types of machine learning things with her MRI images. So this is an issue that's above all of these different physics applications, it's just a general way of autonomous machine learning that I think is going to be a very, very important advance in kind of big data analysis in the 21st century. And we're really interested in those types of algorithms. So if you're interested in that stuff, let me know, because we're getting into artificial neural networks now, and there's always another new algorithm that performs better and better and better, and we have to learn about it. So we're very interested in those things. So as a result, we have shown that, what the net result is, with about a 90 to 97% specificity, 90 to 93% sensitivity, true positives, true negatives, we're able to discriminate these bacteria from each other. So it works really, really well. We've shown that that fingerprint from bacteria is robust and reliable because we take experiments that go actually years in my previous lab. That data was accumulated over years, and it's all very repeatable, and it's great. We've shown that it doesn't matter what the bacteria are feeding on. It still has the same pattern. We've shown that it's independent of how old the bacteria are, whether it's been inactivated by UV light, capable of being discriminated from strains. I think we can get it from about 500 bacterial cells if we really optimize our system. So there's a lot of pluses, and there's challenges too, but mostly everything works pretty well. So I'm out of time, so let me conclude there. My opinion is that this LIBS in general provides an accurate, fast, highly spatially resolved, possibly remote spectrochemical analysis of almost any type of target, solids, liquids, gases, or powders. There's a high degree of versatility to this technique. So what I try to do is train all of my students. They might not be working with bacteria for the rest of their careers, but this idea of using lasers to interrogate something, understanding spectrometers, understanding optics, fiber optic collection, signal to noise, repeatability of measurements, makes this, it's that versatility, because you could use it for almost any discipline. It's going to bring, this is why it's being adapted in many different interdisciplinary fields, including microbiology and medicine. But like I said, geology, zoology, food safety, archaeology, paint and art history, undersea exploration, alloys, metal, the nuclear industry. I could go on and on. It's so versatile, and any of my students could move into any of those fields immediately, because usually those people contact me and say, hey, we'd like to learn about this LIBS thing. Say, my students know all about it. I love LIBS right now because it's an exciting mixture of physics, which I am, laser science, which I am, analytic chemistry, which I'm not, but I'm learning about. And that's just at a minimum. You throw in machine learning, and then there's computer science, certainly spectroscopy. So it's really interdisciplinary. I have to have knowledge of all these different fields, and it keeps me very invigorated because there's always something new to learn, which I love. So I have to acknowledge the people who've been funding me. Canada has been so great to me since I've been here. I love working in Canada because I've been funded really well by the NSERC, National Science and Engineering Research Council Canada. Much thanks to them. A Canada Foundation for Innovation grant helped me buy one of those whole tables of optics and lasers, and that was matched by an Ontario Research Fund. University of Windsor Outstanding Scholars Program supports my students every year. The UWindsor Faculty of Science gave me a very nice startup to help buy my equipment from Wayne State and bring it across the border. All credit to me, is for the students. I don't even do these experiments anymore. I haven't shot a laser spectrum, I would say, in 10 years maybe. It's the students. They do all the work. So when I say all credit to the students, that's not exaggeration. They do all of the work. I'm just there to help them solve problems. So my great graduate students of the past, and Dylan, and this is Russell, and Allie, and Dan, who came back and helped me for a little while. My undergraduates who helped, Derek, and I had Siddharth was a MyTax student from India who visited for a summer, and Khadijah, my very first student, and Laura, and Anthony. This is, my last year's group. So Jeremy and Sydney, who helped out, so much with that video. Thanks, guys. And Paul, who's now graduated, and Allie, and Emma, and Chris. Computer science, we got physics and computer science people, and physics and math people, and medical physics people, and straight physics people. This is my current group. I love to take a group picture every year. This is the first year where I have not been able to get together to take a group picture, but we took a group Zoom picture. So here is my group this year. We are looking for more people. I hope you can join the Rehse group, and you too will get to be a face in a Zoom screen at some point. But we do look forward to meeting back together in person, where we can be shooting stuff again with our laser. So again, these are the people who are doing all my work right now, and they've been working really hard this year and all summer long to keep the progress coming in. So I'm really appreciative of their efforts. So I would say, if anyone on the call who's interested in graduate studies with us, or anyone at the University of Windsor, please just go to YouTube and just search for UWindsor Physics Research. There's a wonderful video on there of all the professors talking about what they do, and then a bunch of our students talking about what physics research has meant for them and their career. It's really good to hear that, and I think that'll really give you an idea of what doing research with us at the University of Windsor would be like. If you're interested in my group specifically, again, I think while you're on YouTube anyways, well, you're watching this on YouTube, it's right there. Why don't you just go up to that search bar up there, type in UWindsor Physics Rehse, you're gonna see this video. That might get you intrigued by what we do. And if you wanna learn more, you can contact me after that. But that'll at least give you an understanding of what it is that we're doing. So with that, I'll end and say, thank you very much for listening. I appreciate your time very, very much, the time we got to spend together. So I guess there's time for some questions now, but thanks so much.
So before we jump into questions, I'll just thank Dr. Rehse again for his talk today. I want to remind everybody, next week, Friday, December 4th at 4 p.m., we have another talk. Next week's topic is new physics and atomic Coulomb explosions following nuclear beta decay. So at this point, I'll open the floor to questions. Feel free to put your questions in the chat.
And I can say thanks to Griffin too. So for people who are watching at home, you might note down in the lower left-hand corner there's our faithful host for the seminar series today. So Griffin has been starting to do some work with me over the summer. And what a great time to start doing research at the outbreak of a pandemic, but we have been finding our ways. Okay, there's a
question in the chat here.
Yep, I can see that. So the question is why did you choose 5 microns and 4.5 microns? Why those numbers? So these are, I'm assuming this question is referring to the filters that we're using. Yeah, these guys, these are commercial filters that we buy. This is a company called Millipore. So these are standard chemical or microbiological separation filters. So we have to get the pore size that they produce. And so that's really all they have. There's a 5 micron, there's a 0.45 micron, there's a 0.22 micron. Those are the standard sizes. So that's why we choose. But it's a good question because we were specifically, maybe that question is actually a bit deeper than I thought, because we did give it some thought. When you get like a blood sample or any other sample, what we are trying to do is separate out the bacterial cells, which are called prokaryotic cells, from the human cells, which would be eukaryotic cells. Or do I have that backwards? See, I'm not a microbiologist, which just goes to show it right there. There's human cells and there's bacterial cells. And the human cells are much, much bigger, right? Those red blood cells are all about 10 micron in diameter, and they're about the smallest of the human cells. So we did pick a size. So that was intentional, right? We pick a size that will catch something that's 10 micron, but allow a bacterium, and a bacteria is about 1 micron in size. Our E. coli that we sample is about 1 micron in size. The 1 micron will go through that filter, but the 1 micron cannot go through the 0.45 micron filter. So while those are the commercially available sizes, they were carefully chosen to do that specific separation. So that was our target. Now, we have not started working with blood yet, but I think that's what we're going to have to move into. For a while, I was worried about the biosafety restrictions, but I think we can just do studies on sheep blood. So you don't have to have like human blood donors, but you can just buy sheep blood commercially. And then we'll add in bacteria and pretend it's a simulation of a human infection. I can honestly tell everyone on the call that when I give this talk, when I talk to doctors, they get really excited. And what the doctors really want to know is blood. They're like, can you do blood? Can you do blood? Blood is a big, big deal for them. Blood infections are no joke. Yeah, well, the platelets for sure. That's why we have to do the experiment and see. So the question is are you worried about platelet contamination? Yeah, there's other types of stuff in blood too as well. That's going to be the problem because it's not just cells. There's actually stuff dissolved in the blood, right? There's stuff in solution. And if we catch that, we're going to see those things as well. That's why, to be completely honest, why I've stayed away from blood. It was not the biosafety business, because we can always get biosafety permission like we have for the bacteria. Blood is a very complicated matrix because it's got platelets and cells, but all these dissolved chemicals and other stuff. So that's going to be what I call a hard problem. And I'm trying to concentrate on the easy problem. So even when I was at Wayne State, a better problem is a urinary tract infection. Now, urinary tract infections or UTIs are not nearly as dangerous because they kill almost nobody as sepsis. On the other hand, it's a question of scale. The number of sepsis cases per year is small. The number of UTIs per year is in the millions. It is literally millions. And so the ability to diagnose those infections very, very quickly, and your urine is sterile and there's not stuff floating around in it. So that was a test that we actually did simulate. We had a paper on that of simulating urinary tract infection, and it worked really, really well. So I'm worried about all kinds of contamination in blood, which is why we've avoided it, because we're still working out the kinks in our new ways of doing it, and blood will be very difficult. So when the doctors say, can you do blood? Can you do blood? I'm always like, I'm hesitant. Put it this way. If I had four postdocs working with me, one of them would be working on blood for sure, right? But because I usually have a smaller number of graduate students, we focus on what we know will kind of make real progress in a short time period. Blood's like a what do you call it? It's not pie in the sky, but we'd have to reach for that a bit, and it might not work real well. So blood would be great. But I hope people on the call can realize, I mean, I think when Jeremy does his defense next week, he gives a really good idea of showing that this idea, like even just what I'm showing on the screen right now. So we're putting a fluid sample through a dual centrifugation device to get the big stuff out and the bacteria collected. But this is a medical application, but this could be used in a food processing application. This could be used in a water safety application. So there's environmental applications. There's food preparation and beverage preparation and biomedical. And between those three fields, that accounts for like over $2 billion of research annually into how to do these types of things. And our one technique solves all of them. That's what we think is so great about it. The exact same protocol could be used to identify contaminated water coming out of the Detroit River. It could be used to identify contaminated cantaloupes coming off the food line. It could be used to identify contaminated blood in a patient. That is tremendous flexibility. That's what we're chasing after. it's really cool. It's really exciting, but it's a big project. We work on it. I said we always have work to do. Yeah, but it's cool.
Well, if there's no further questions, I'll thank you once again, Dr. Rehse, for your talk today. If anyone has any further questions, feel free to email physics@uwindsor.ca and we can connect you with somebody who might have an answer. And I will thank you for attending our talk today. It will be available on YouTube on the UWindsor Physics channel in the next couple of days. Thank you.
All right, thanks everybody for coming. I very much appreciate your attendance. So, and thank you very much, Griffin, for agreeing.